Okay, can you see my
Yeah,
screen?
yeah, we can see the screen.
Oh, great.
Yes.
Okay, so I guess I should do the slideshow from the beginning. Okay, so I think I should disclose that I'm a well, this is the first slide. I'm going to talk a little bit about a little bit of an update on disulfiram, and then a little bit of time about the NIST membrane detection method. But I also think I ought to disclose that I am an investor in Convicure. I'd like to take a minute just to talk about that. I first met Jayakumar Rajadas at a meeting at Mass General in 2015, in which Nev Zupčević, who later headed the uh, the Lyme Evaluation Center at Spalding Rehab Hospital in I think it's in Charlestown, Mass. She and Elizabeth Leandrowski set up this meeting in which the previously warring sides in the Lyme Wars that were all together under one roof, and it was actually very convivial. We all got along and shared, and then I met Jack Kumar at the cocktail party afterwards. And since that time, I've gotten to know him well, and I'm very impressed with his creativity and innovation as a scientist, and the great work that he's done. And so I've gotten to know him, and I also realized that his commitment to Lyme disease patients, in terms of trying to develop improved methods of treatment, it's I really we were talking about it. It's really a calling. And Anyway, I'm a pretty cautious guy, but I decided, hey, put your money where your mouth is. So I decided to invest in CombiCure. This is a slide that I was fortunate to get. I first saw this slide that was given during a talk that Willy Bergdorfer had given. I forget, it was at one of the Lyme Disease Foundation talks. And it's just a great slide. And it was created by a graphic artist who was out at the Rocky Mountain Laboratory. I recently contacted Tom Schwan to see if he had access to this slide, and he kindly shared it with me. And this battle is still going on, man versus ticks and tick-borne diseases. I just love the slides. People can appreciate it. I'm pretty sure it's out there in the public domain if anybody else wants to use it. Anyway, I've given many talks about disulfiram since our first paper came out in around 2019. Um, and I'm not going to go into as much detail as I might. I believe, Jaya Kumar, you're going to make available to people who want the PDFs of those articles, right? So anybody who wants to go in greater depth can delve through those.
Yes, I will do
Um,
that. Yes.
yeah, so I was vaguely aware that that article, but really what brought it to everybody's attention was at the Lyme Mine conference at Mount Sinai School of Medicine, which at the time I wasn't even aware it was happening, but it was recorded and put out there on YouTube. And during the talk, the keynote speaker was Kim Lewis, who's doing great work. He's up at Northeastern. And he commented that one of the most potent agents against Lyme disease had been discovered by Dr. Rajadas and Dr. Potaneni in their high throughput studies of drugs that were also already in the pharmacopoeia, and it turned out that disulfiram was way at the top, very very potent. So one of my patients who was following Kim Lewis's work, happened to see that YouTube, brought the article to my attention, and I reread it because I had seen it originally. And he says to me, "Hey doc, can I try this stuff?" So he was a very reliable patient, after giving it a lot of thought, reviewing it, reading up a little bit on disulfiram, which I had never used because it's traditionally been used in treating people with alcohol problems. I decided, and he was a very well documented case. He had struggled with Lyme, was very well documented, and he did respond. Anyway, this is Ravi Potaneni and Dr. Rajadas. And this was the article in which I guess they tried thousands of different candidates against Borrelia in the test tube. And this is the structure of disulfiram. It's actually not a very complicated molecule. Doesn't have a very high molecular weight. And anyway, so that first patient persuaded me because I trust me, I never would have suggested it to a patient. It was only the patient suggested it to me, and he'd been around the block many times with different kinds of antimicrobials, and he did respond. But if you stop treating, he would relapse, and that's rather demoralizing. So I decided to let him try it, and he was the very first patient who was on it, and he had a very gratifying response. He went into remission, and so that was the first patient. And then, encouraged by that experience, I mentioned that to a couple of other of my patients who were all very, very well documented. I'd worked with them for years, and they needed very intensive treatment to keep their heads above water. So I tried it in two other patients, and like I jokingly say, bing, 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 all three of them entered an extended remission without need for further treatment. And so that was so striking to me that I practically, pinched myself and I said to my nurse, Dawn, we have to report this because it was so striking. And that led to our very first paper, which is just the report of these first three patients. And the amazing thing about that was that it was one of the few opportunities in the medical literature that we had, thankfully, to the editors at MDPI Antibiotics. They let us report in depth the clinical histories of these patients, which were, at first I thought, well, I'll just do a kind of a paragraph summary. Said, no, these patients, I have to let people know what these patients had gone through. So again, I'm very, very appreciative that the editors allowed us to publish that and have these very extended case histories detailed with everything that they had gone through. But anyway, so that was the first three patients. I'm not going to go into the details. This PowerPoint will be available for people to review, or it'll be on YouTube, right? So I'm not going to take too much time. This tells you what we did with him, and he was on 3 months of disulfiram. There was a second case, and this happened to be, I think this was a physician, and required very, very extensive care. And he also did the disulfiram, and also he had a syncopal episode. But anyway, he had a very good result. And then a third case, a patient that I'd worked with for many, many years, and also had an excellent response. Anyway, I'm not going to go into the details, but they're there if anybody wants to look at them. And then subsequently to these three patients, over the next year or two, we applied disulfiram. I had collected a lot of the patients who had chronic or long-term Lyme, and so given the response of these three patients, we offered them to many of our patients, and as a result, we had a cohort of about 67 patients that ultimately were reported on because a few were lost to follow-up. But and then I summarized the response with them, and it was an observational study. It was not a placebo-controlled double-blind study. And also, I didn't feel that I needed IRB approval because this was just use of a already FDA-approved repurposed drug on clinical patients. And we reported our experience. And overall, it was very positive. We retrospectively analyzed these cases and found out that, geez, some of them we had used a fairly high dose, and some of them we used a fairly low dose, and we used a cutoff, I think, of 4 milligrams per kilogram to separate the high and low dose. And we found that with a high dose, about a little bit more than a third of those patients, I think it was like 36%, were able to enjoy a long-term remission, which we defined as 6 months or longer without other application of other antimicrobials. Some of those people relapsed, but some didn't. Actually, I was in touch with one of my patients who was acquainted with one of the people who was in that study who we'd lost contact with. And I just asked her if she could reach out and see how he was doing. So this is like, 4 years later, and he's well. So, I can't say that happens with everybody. I don't think this is a magical cure or a panacea, but for some people, it's has been very, very effective. And some people did have problems, and we reported the kinds of problems that we had. Fortunately, in the patients that we took care of, who we followed very, very closely, no one had any kind of a catastrophic um, problem as a result of the use of disulfiram. But it should be known that disulfiram has been associated in the past with significant problems when it was used for alcoholism. And also, I should mention that in the early days of its use, um, much higher doses were typically used, like between 1 and 3 grams a day, which are huge doses. So there is an extensive literature of potential adverse effects of disulfiram. And I also think that any physician who applies disulfiram in his patients needs to really familiarize themselves with the agent. I went to some length to discuss adverse effects in my papers, especially the first one, because I think it's very important for both the physicians to be aware and also any patients who might undergo it to also be aware. And that although it's very useful, it has to be used with great care. And so I think I've emphasized that in my paper. Um, Also, at the time that the two papers were published, I hadn't had enough experience or follow up with the patients who were on low dose um, to be able to know whether or not, with a low dose approach, any of them could enjoy an extended remission. But since that time, we've learned that some of our patients who are on quite low doses were able to stop and remain clinically well. Um, and I go into some of the different kinds of side effects that we encountered. And again, in all of them, they were reversible. But some people had psychiatric symptoms. Some of them, a small number of them, really required involvement, in psychiatric care, which was, temporary and short-lived. Um, and some people had fatigue. A number of patients developed neuropathy, most of whom, but not all of whom, was reversible when you stopped the drug. Um, and okay, so let's see. Then we it seemed to us that for a lot of the patients, the symptoms of babesiosis also seem to go into remission, although that's much less well characterized presently than its status in its effects on Borrelia. And then also with Bartonella, a fair number of our patients had Bartonellosis as well, and we were really not able to determine that it had a clear impact on Bartonellosis. And a lot of those patients who had Bartonellosis opted to be concurrently treated with more standard treatments for Bartonella, like, for example, zithromycin, minocycline, rifampin. So it was hard for us to really tease apart. I think it's my impression and that of many of my colleagues that disulfiram is not particularly active against Bartonellosis. Um, as far as the perils, it's well known that disulfiram can have adverse effect on the nervous system, both peripheral nervous system and also even on the central nervous system. And this is some work that was done out of Vanderbilt. I think the chief scientist was William Valentine, who did really some excellent studies with his colleagues. This is one of them. This is in a rat model, showing that disulfiram can produce damage to the myelinated fibers in the rat sciatic nerve. On the top, you see the myelinated fibers with the normal myelin sheath. And towards the bottom, you can see how disulfiram in this model can really damage the peripheral nerves. And that's very, very important for practitioners to be aware of. And if you start seeing evidence of that in the patients, what they report to you, it's mandatory to discontinue the disulfiram. And not only can it have adverse effects on the peripheral nervous system, but also can have adverse effects on the central nervous system. This was a case that was reported, an unfortunate case that was reported in Mayo Clinic proceedings. This is a case out of Pennsylvania where the patient, I guess, did not follow advice of the physician, used higher doses than was supposed to, and also was on concurrently was on another drug that is contraindicated with the use of disulfiram, was tinidazole, and he ended up having damage to the brainstem centers that ultimately led to a fatal outcome. Unfortunately, the authors took this as an opportunity to attack the whole notion of Lyme as a persistent infection and attack the physician to take care of the patients. So, but that was their axe to grind. But it was a case, and this type of event had been reported already in the literature of its use for in alcoholism. As far as the mechanism of action, I don't know if Dr. Rajadas and your colleagues, whether you've been able to really pin down the exact mechanism, the beneficial mechanism against Borrelia. I have some speculations about it, but I still don't know if we have it exactly pinned down. Maybe Dr. Patula can talk about that. I've listed some of the things that have occurred to me as possibilities, but again, there's speculation. I also want to mention that it's now well documented that Borrelia can produce biofilm, not only in the test tube but also in humans. This was and this happens to be in vitro. Eva Shapi used atomic force microscopy to show the structure of biofilm. You can see those pores. The biofilm really has a structure, and it's produced by the organisms that reside within it as a protective mechanism. It produces this kind of slimy layer, but it tends to protect organisms within. And in fact, Eva told me that bacteria within biofilm are 1,000-fold resistant to the action of antibiotics. One of the speculations that I've made about, but these are biofilm that were detected in a fatal case of mine, the famous case of Vicky Logan, in multiple tissues. Using special methods, Ava was able to document large biofilm clusters.
Ja, das ist ja schön.
Some of these are as large as 300 microns in diameter. The green on the left shows biofilm in heart, kidney, liver, and brain as well. But the point that I wanted to get to, first of all, disulfiram itself is a fairly small molecule, but furthermore, one of its breakdown products, carbon disulfide, is about the size, which is produced in nanomolar amounts from the metabolism of disulfiram. And it's a very small molecule. You'll see that that oxygen and sulfur are very sulfide are close to one another on the periodic table. And in terms of its size, carbon dioxide and carbon disulfide this is in picometers, are about the same size. And so again, I've speculated that, and carbon disulfide does have antimicrobial properties. So I've speculated that might be one of the means by which disulfiram has activity and benefit. Okay, let's see. Yeah, I just want to say that since 2019, we've explored the parameters with which you can use disulfiram. I've had one of my patients was on 100 milligrams 3 times a week. She weighed about 98 pounds for more than 4 years with benefit and without obvious toxicity. And one of my patients, he was actually one of the patients in the first 3 that I reported. He subsequently had tick bites and got Lyme disease again. But he's been on 500 milligrams 5 consecutive days a week, once a month, for a couple of years now, with excellent results and very good quality of life. So I think the parameters are still being defined. This is our team who reported on that paper. This all happened during COVID time. And this is us without masks and socially distanced. And I provide extensive references, which I would encourage people to explore. And with that, I think we'll take a few minutes for Q&A.
Yeah, audience are welcome to. Thank you very much, Dr. Legner. Yeah, audience are welcome to ask questions if they have any.
Is there using the chat function, or are they going to verbally
No,
ask
they, they,
questions?
they are going to verbally ask
Oh,
questions.
okay.
Dr. Kenny Kleiner, that was a very good talk.
Thank you.
So yeah, like the question what I have is about the brain fill study, which they did the clinical trial. So do you have any update on that? Because I was trying to get into because they had recruited 11 patients and they were saying that the fatigue is the one of the issue. But do you know any in detail about like what was the like outcome of the study?
I do not know. As far as I know, it still has not been published, but I believe there is an intention to publish it. I don't know why it's been a long time. I don't know why it hasn't been published yet, but I believe that, of course, it was also interfered with by the pandemic. It interfered with their recruiting. But I believe they have patient experience, and as far as I know, it's Dr. Fallon's intention to publish it, but I'm not sure when, and I don't know the details.
So I have a very another question about like your study. You have the low dose group, or you have just only the like low dose and high dose? Like, is there have any variation in the dose? Because I haven't. Maybe I missed out the slide that one.
What's the question again, Dr. Pochulo?
So do you have a low dose and high dose patient?
Oh, well, we used we analyzed what we actually did with these patients, and we each of them were individualized. We didn't have like a cookbook, and then
Okay.
we looked backwards and we saw, well, the high dose was usually above 4 milligrams per kilogram, so between 4 to 6 milligrams per kilogram, and the low dose was below that, and some of them were like ridiculously low. We had one patient, quite well characterized. He did only 62.5 milligrams once a week for a number of months. I think it was maybe 10 months. And with that, he was able to stop. And as far as I know, he's been rather well. I think once in a while he'll take some doxycycline or a little bit of Malarone, but he's as far as I know, which is like practically homeopathic doses if you think about it, like a quarter of a 250 milligram tablet once a week. But what can I say? It seemed to really help him. So anywhere between 4 milligrams per kilogram per day and 62.5 milligrams. Well, that was just, I don't know what the milligram per kilogram would be, but just that's the lowest dose that we saw benefit with. It gives you a notion of how powerful this drug can be for some people.
Great, thank you.
I have used disulfiram for quite long, but I stopped 3 years ago. And my hypothesis is that my Lyme is not completely gone, but my defense system is coping with it. So I don't need medicines anymore to suppress it. That's my feeling about illness, because I have migratory headaches still and some muscle twitches, which for me point at still an active infection, but it's very very good without medicine to live. Very, very good life quality. I am excellent in my job nowadays. I've promoted, so I doubt anyone will see I've ever had the illness, but I know it's still there.
Well, it's a useful tool to have in the armamentarium. Personally, I don't generally use it as like a first line thing. I mean, it's still not considered, I wouldn't say it's considered, quote, standard of care. And for most of the patients that I deal with, we go through, more standard approaches. And I usually only resort to it in my patients if there's a compelling reason to use it in a particular case.
Yeah, I had psychosis after the first try, so it's quite risky.
It has to be used with care for sure.
I had a question, Dr. Lingner. I am currently on my second round of disulfiram. I used it in 2020 with great success. I had 3+ years of what I call pretty complete remission. It was the first time in many, many years I've been back and forth with, antibiotics and so forth since 2007. So the disulfiram really is what kind of kicked it for me. I, at that point, had had a new infection to include Rocky Mountain spotted fever, and I had already treated previously before the disulfiram was introduced with antibiotics. Unfortunately, I was a second-time person who got C. diff, so antibiotics are really no longer an option for me. So then we did the disulfiram, which did really great until this. I just got sick again this past November. Looks like I had COVID, turned long COVID. And my doctor treated, with high dose ivermectin for the long COVID, and then decided my Lyme must have been reenacted. I don't have a new bite. So I'm back on disulfiram very, very freshly, just started actually this past week. And I guess the question that I had for my doctor and also for anybody who's dealt with disulfiram quite to the level that you have is it typical for somebody who doesn't have Lyme or co-infections to have a severe reaction? Myself personally, I'm medication sensitive, so I have it compounded through a pharmacy, and I was to start at 50 milligrams. I accidentally started with the wrong strength. I started with 100 milligrams. Within hours of the first dose, I had a pretty significant headache. And within, by the next day, I had an unbearable headache that I couldn't treat that turned migraine. So I feel that I definitely reacted to the medication. And my question is this a normal reaction for somebody who isn't like an alcoholic, somebody who doesn't have Lyme or co-infection? Is it a typical reaction to the medication just because it's such a, as great as it can do, it's a scary medication too?
Well, I have to tell you, I don't have any experience using disulfiram in the treatment of alcoholism.
Right.
So I don't really know. As far as I can tell, from what I've spoken to colleagues who do have extensive experience having used disulfiram in alcoholic or patients with alcoholism, and as far as I know, they did not have anything that would be suggestive of a Jarisch-Herxheimer type reaction. Most patients tolerated pretty well, and unless they had major toxicity, which could happen down the line, and with high doses.
Well, I guess I'm just thinking that's the only reason you would have that reaction, right? It would have to be that's what's going on, right? Is that like the testing can be inaccurate, as I'm sure you're well aware of with Lyme disease, in the various testing facilities. Some are better than others for sure, but if you're not getting a conclusive test and then you try this, is that a way to kind of tease it out to know that, yep, this is Lyme we're treating?
Well, life is complicated. Patients are complicated. I wouldn't want to go so far as to say that you could use it as a diagnostic test. Although, of course, many people realize with antibiotics, for example, when you apply antibiotics and people have characteristic, multi-system symptoms that are consistent with Lyme, many people take that as a sort of a supportive that they may be dealing with an active, Lyme infection. So the same thing, I guess, could be true of disulfiram, but I wouldn't want to advocate it as being a precise diagnostic method, but it's possible.
All right.
Sure.
So there is a question by Barbara Marcus. So you wanted to ask yourself, Barbara?
Yes, I did take disulfiram. Actually, it's been a couple of years. It took me several months to ramp up to the appropriate dose. I continued taking rifampin during the time that I took the disulfiram. I got up to the recommended dose for my height and weight and remained on it for about 3 months. I did see a significant improvement in the Lyme symptoms. However, I also have Bartonella and saw a pretty big surge in those symptoms, including face and head pain, neck pain, shin pain, bottom soles of feet pain, muscle pain, etc. So at that time, I decided, because the disulfiram was also affecting my GI, to go off of the disulfiram. Are you seeing that, I'm hearing on a lot of the, Facebook support groups, et cetera, that a lot of people are showing this surge in Bartonella symptoms when they're using disulfiram? Are you seeing that to be a common reported, symptom surge with Bartonella? It did seem to be outside of my normal, I guess, symptoms, and it was not well managed by the Bartonella. I mean, I'm sorry, by the rifampin at that time either.
Usually rifampin is not recommended as a monotherapy for Bartonella. It's usually combined with other agents such as the
Yes.
azelides or a tetracycline. But
Yes,
anyway,
I was taking tetracycline as well. I'm sorry.
but anyway, I mean, yes, that was an observation that that we found. Like I said, some of my patients with Bartonella opted to continue their Bartonella treatment, and some of them opted to just do the monotherapy with disulfiram. And I think we observed, and other patients and other people have observed, because disulfiram is really not treating the Bartonella. So that, in a way, if you don't concurrently treat the Bartonella, if the person has Bartonella, then it gives Bartonella an opportunity to be untreated for a while and perhaps become more obvious. Some of my colleagues have combined disulfiram with methylene blue. And of course, neither of them are antibiotics or antimicrobials in the usual sense. And also, neither of them, as far as I know, I mean, I'm not an expert with methylene blue, but neither of them, as far as I know, have a big impact on the microbiome, you know? And so a number of my colleagues have been very upbeat about using disulfiram and methylene blue in combination. Tom Moorcroft is one of my colleagues in ILADS, and he's given presentations and talks on that combined use and about methylene blue itself. But yeah, I mean, that's one of the things that makes it so challenging caring for patients with tick-borne illnesses. Many of them do not have a single infection, but sometimes multiple, 2, 3, or even more. And we jokingly talk about the 3 big Bs: Borrelia, Babesia, and Bartonella. And an awful lot of our patients are turning up with Bartonella. The CDC still is insisting there's no, good proof that ticks transmit Bartonella, but some people disagree with that, and an awful lot of people with other tick-borne illnesses turn up with Bartonella as well. So I think the jury is still out about whether Bartonella is tick transmissible. It's certainly flea transmissible, cat scratch transmissible, and we know that vets, we know that for veterinarians, Bartonella is considered an occupational hazard. They're dealing with dogs and cats. They have exposures to fleas and whatnot. So yeah, it's challenging. All of these are challenging, and I've jokingly said taking care of these illnesses for the physician, it's like playing 3 simultaneous games of chess against 3 chess grandmasters. Our opponents, the infections. It's very challenging for the best of us.
Thank you.
You so much, Dr. Ken. Maybe we can proceed with the next talk. And if people have questions, please send us an email, and I will just forward the emails to the speakers, and then we can get the information whatever you are waiting for. Thank you.
Sure, I'll be happy to try to answer them if I can. All right, so let's proceed then. I want to talk a little bit about this. I am so frustrated that there's a method of direct detection for Lyme, and it's sitting on a shelf. And I've tried to promote the notion. I've talked to a lot of different people, and there's no takers on it. And it's just, let me talk a little about this. And I don't know whether it's something that Convicure could take on or people would want to take on, but well, let me just go. So NIST stands for the National Institutes of Standards and Technology. And what happened was John Aucott, who's done some great work at Hopkins studying Lyme disease, and people probably know about his so-called SLICE method. I'm not sure what the acronym stands for, but he took patients with well-documented erythema migrans, positive Lyme patients, and studied them in tremendous detail. And he's published many articles about that. But the thing is, at a certain point, he took specimens from those patients and shared it with researchers at the National Institute of Standards and Technology. And John said to the scientists, and the scientists looked at the data, and everybody agreed, yeah, our tests for Lyme are lousy. So these workers at NIST tried to see if they could come up with better methods, okay? And as a segue into this, many, many years ago, researchers at CDC asked me to provide them with specimens from the patients that I was caring for, because they knew that I had patients who either might have Lyme or did have Lyme. This was early in the late 80s, early 90s. And so I sent a bunch of specimens of serum, urine, and spinal fluid to folks out at CDC in Fort Collins. And unbeknownst to me, they forwarded them on to people at the Rocky Mountain Laboratory, Claude Garan, Dave Dorward, and Dr. Schwann, Tom Schwann. And again, unbeknownst to me, they had developed this method of detecting the blebs that are shed by Lyme spirochete. So Borrelia, as part of their biology that they can't avoid, they shed these lipid-laden blebs. And those blebs, by the way, contain genetic material. It's probably part of the life cycle of the Borrelia. So they developed this method. It's published in the Journal of Clinical Microbiology, which is like, a premier journal. The only thing about this method was that in order to use this method, it required use of electron microscopy with an immunogold label that was able to detect these blebs. So the thing about that is, that's very, very labor intensive, not at all suitable for commercial purposes, for, high throughput purposes. It's just too difficult. Anyway, so it turns out they studied specimens of urine and serum from 51 of my patients, all who had been well characterized. And then Claude Garan gave me a printout summarizing the results using their direct detection antigen capture method in the urine of my patients, and then compared it with 3 different Lyme antibody tests that had been formulated by folks at CDC Fort Collins. So this is the printout of the results. And in the first column is the RMLs for the Rocky Mountain antigen capture assay. And then there were 3 different ELISAs. That ELISA TG stood for TQ stood for Tom Kwan, who was one of the scientists out there. But the thing that was most striking, if you look at this column, there were 37 patients who had a positive antigen capture study in urine, and they were dead negative by 3 different antibody tests. So that was one of the early clues to me that our tests for Lyme are grossly inadequate, and made me open to realizing that many patients with Lyme could test negative on the standard tests, but they still might have Lyme by direct detection. So it turns out that Richard Tilton, who actually he was the editor of the Journal of Clinical Microbiology at a certain point, he had his own laboratory, BBI Clinical Labs and North American Lab Group. He actually got the license from the NIH to try to commercialize this, and he's passed away now, but he had confided in me that he'd spent about $1 million trying to adapt this method to something that would be suitable for high throughput commercial sales. He finally gave the license back to the NIH because he just couldn't do it. Okay. And also another thing, I had other access to research assays at the State University of New York at Stony Brook, where they had developed direct detection methods for outer surface protein A and outer surface protein C. Again, these are shed on the membranes of the organism, and also Borrelia-specific immune complexes. And I just want to point out that using standard assays in spinal fluid, these are results of spinal fluid from my patients, well-characterized patients. Only 2% of them tested positive on the standard antibody assays, but 61% of the specimens tested positive on one or more of, there were 4 different research assays, including those 2 direct detection assays. So again, another indication that our generally available methods are grossly inadequate. So this is the manuscript that was published. It's open access. Anybody can access it, about the method that the people at NIST detected, which used multiple reaction monitoring, liquid chromatography, and triple quadrupole tandem mass spectrometry to detect specific membrane outer surface proteins shed by the Lyme organism in these specimens that John Aucott provided to them. And this was sensitive down to the femtomole level. So, Jai Kumar, maybe you can explain to people how sensitive that is. But anyway, and I actually communicated with the lead author. His name is Ilarion Turco. I communicated with him and I said, is there some reason this is just not feasible? Is the method too difficult, or is the equipment too expensive, or it's not suitable for high throughput, or maybe it's patent protected? And he said, no, none of those things are true. And furthermore, he told me, he made a point, this is not patented. This is in the public domain for anybody to use. And that's why I'm so frustrated. Nobody's using it. So I believe that the NIST method could provide an objective, reproducible, qualitative direct detection method for assessing the presence of Lyme infection, and furthermore, a semi-quantitative means of assessing the body burden of infection, or I use the term borrelial load, analogous to the viral load in assessing HIV infection. And and this is based on the fundamental biology of Borrelia. So why isn't it being, adopted by anybody? The other thing is that this membrane detection method could be adapted to other infections, including, for example, syphilis. It's well known that even the CDC is desperate for better methods of direct detection for syphilis, that you could better characterize the stage of illness. They're still depending on these archaic methods of following the titer of the VDRL, the RPR, and spinal fluid to tell, is the infection progressing or not. So, I mean, there is PCR, but that's very low yield. Anyway, is there a path forward for the NIST method? It would require substantial funding, probably at least $5 to $10 million to bring it to fruition. So my concept, but I don't know whether it's feasible, and this is not what I do, so I don't know I have no clue how you would do this, but perhaps have a startup company with private investors, and then the attraction of a venture capital might be a viable path forward. It would need to be, studied in carefully characterized patients and studied under conditions of treatment and observation to see whether it really correlates and whether it's useful. But again, I know there's this, the Cohen Foundation has this X Prize to try to expedite direct detection methods. But as far as I know, none of those methods that are under consideration are using what is so obvious to me and so fundamentally based on the basic biology of the Lyme organism. Anyway, that's my pitch. I don't know whether it's something that Convicure could take on or not, but I would love it, and I would. It's not something that I have the capability of doing, like personally. It would require, a lot of people cooperating in order to bring this to fruition. But again, this is what we've needed, and I don't understand why CDC isn't jumping on this, NIH isn't jumping on this. It's almost like nobody really wants to know, because you might be able to find a wide range of illnesses that are presently obscure as to etiology, that could be correlated to borrelial infection and perhaps other spirochetal. Oh yeah, I'm going to shut up. That's all I have to say. I did provide some references for that as well. And this is an abstract that Claude Garan, myself, and Dave Dorwood presented in the 5th International Congress on Lyme borreliosis about what we found with the patients that we had been able to study. And I was one of the few clinicians fortunate to be able to have my patient specimen studied using this antigen capture assay that was developed by some of the finest scientists in the world at the Rocky Mountain Laboratory. Okay, I guess we can take some further questions.
Thank you, Dr. Lechner. Yes, go ahead. Go ahead. I have few comments, Ken. Again, my name is Jaykumar Rajadas, and so my comment is like regarding the expensive. Actually, I by looking at the protocol with all the technology development, I don't think it will be that expensive. Actually, it is somebody needs to take that up. Like there are so much patient need. Perhaps one of the philanthropists or somebody, they need to consider promoting it, and then putting some money and then trying to find out whether they can help people. That's my take on that.
Well, it seems to me that if I'm right about its utility, it would enable people to objectively measure is our treatment effective in decreasing borrelial load or not. So not only for diagnosis but also for guiding therapy, it could be so useful. And again, I'm incredibly frustrated that it hasn't been brought to fruition. Maybe for a lot of people, this is the first time that it was ever brought to their awareness that this method has been developed. So trying to get the word out there. And so I actually had made a proposal for a national reference laboratory for Lyme and spirochetal diseases. And I sent that to you, right, Jack Coomer? So you can make that available to people at will.
Yes, I'll do that, yes. So in the absence of any questions and comments, perhaps we can thank you again, Dr. Kenneth Lechner. Thank you so much. And we will proceed with, Dr. Harihara Potula's thing.
Yeah, thank you, Chris. Can you see my screen?
Yep.
Okay, all right. So before I talk about my topic, I just want to give a little bit background about myself. So I started my research in US in 2009 as an immunologist. Mostly to start with, I was working on the cardiovascular and infectious disease. And then in 2014, I ended up working with Maria, who is an expert in vaccine-targeted Lyme disease in University of Tennessee, Memphis. So I've been working with her for almost 2017, where I met with the vaccine research meeting at Vancouver. I think I met Jay in 2017. That's how he invited me to join to his group. Because in Maria's lab, I was working as an assistant research professor, mostly working on the mouse models of the Lyme. And also there is a cousin of Lyme disease, like the Leptospira, which is another spirochete. So I was working on both the models. And I developed both the mouse models in the lab, both, it's not only the straightforward needle-based injection, and I had developed the tick-based method. So that's how actually Dr. When he saw my presentation, Dr. Jay provided me the opportunity. He said, why don't we have a collaboration? Because we have a couple of interesting small molecules we just discovered. At that time, they just discovered a couple of molecules. That's how I ended up coming and started Jay from 2017. And now we are still continuing working with him. So in this topic, actually, I would like to talk basically the little bit introduction about the Lyme and the current status, and how a little bit history of the lab where we had generated a couple of small molecules and their efficacy on the Lyme. And last few slides where I would like to talk about the polymorphic formulations of the, because we end up getting the disulfiram, but again we wanted to have the disulfiram in lives, because as we see the couple of patients and a lot of the review articles, they're saying the doses are pretty much higher and that it needs to be optimized. And that's how we end up trying with different formulations of disulfiram. And those are the couple of the slides which I'm going to talk about at the end. So can I start the presentation now? Okay. Can you see my slides? Okay. So as you all know, this was the first Lyme arthritis. It was an rheumatologist actually who identified an outbreak of arthritis. That's how it initially thought it was in juvenile arthritis. And they discovered in basically the almost 40 children, and they clustered into the small town called the Lyme, so which is in Connecticut. Still, it is in Connecticut. So, as we all know, this is the causative agent is the bacterial species which belongs to the phylum Spirochaetae, and then because it looks like in a wavy body organ or a flagella organs of the mortality, they enclose between the outer and the inner membranes. It was in 1982, Dr. Willie Buckdorf, who successfully isolated this Borrelia, and that's how it has its name as Borrelia burgdorferi. And you can see the SEM images below. And mostly in North America, it's the Borrelia burgdorferi, and the agent which causes is the ticks, which are Ixodes scapularis. Other than that, there are two other species, which are the Borrelia garnier and the Apzili, which are the pathogenic in Europe. And if you can see, these are the different forms of the larval stages, the mold to the females and the males. And I'm going to talk a little bit about in the next couple of slides. And if you see the majority of the Lyme disease cases in the US, they are mostly occurs in the temperate regions of North America, Europe, and the Asia. So in the United States, if you see the prevalence of the reported cases, they are particularly in the distinct geographical pockets. If it is specifically the endemic regions, they kind of mostly can see the northeastern, mid-Atlantic, and north central. I can see from the Maine to the North Carolina, the Michigan to Wisconsin. Historically, it's the Connecticut which has the greatest amount of the reported cases. Here you can see that each dot replaces randomly within the county of the residence for each the confined case. So if you see the life cycle of how this black leg ticks, the Ixodes scapularis and Ixodes fasciculus, which is generally the how it generally lasts for 2 years. If you can see from the nymphal stage, and it again goes back to the eggs. It is kind of 2 year cycle. Actually starts in the spring, goes to the summer with the larval, and then goes to the fall again, and then goes to the winter, and again comes back in the nymphal form. And again, if you see the entire journey, they go through the 4 stages. Starts from the egg to the 6 egg larvae to the 8-legged nymph, and then adult. After the eggs hatch, the ticks must have a blood meal at each and every stage to survive. So the black leg ticks, they can feed on any mammals, like from the birds to the reptiles to the amphibians. And the ticks, they need to have a new host. Like if you can see at the larval stage, they're mostly targeting the birds and the rodents. And then at other stage, they mostly target the deers. So each and every cycle they target, and basically the mammals are the kind of when we go to any camping, so we are not the right targets, but we are like the targets which happen to be targets actually. So the mostly they target the days. So the signs of symptoms, the early signs are the like fever, chills, headaches, fatigue, muscle and joint pain, swollen lymph nodes. They occur in the absence of the rash. You can see this in trauma migrants to start with within a couple of days, from 3 to 30 days with the first tick bite. And the last signs which leads to the chronic is the facial palsy, which you can see on the bottom, which where there is a loss of muscle tone to the droop to one side, or sometimes it happens to the both sides of the face. And then as I like, this travels, this disseminates into three systems. Through the circulatory system, it causes the Lyme carditis. Through the lymphatic system, it goes and settles the joints, and it causes the Lyme arthritis. And through the neural system, it goes and accumulates in the central nervous system, and it causes the neuroborreliasis. So these are the couple of organs it targets. And then before I go in detail about the history of the diagnosis and the treatment, the different antibiotics we started, I just want to show you the current slide. This I just downloaded in the morning today from the CDC, where you can see the estimate that over almost 300,000 people are likely diagnosed and treated each year on an average. And then the number of the cases they kind of steadily increased from the last couple of years. But these 2 years, the 20 to 2021, because of the COVID, they haven't reported complete cases. But you can see, and there is a steady increase over the past couple of years. So there is a required for the new diagnosis and the treatment, as Dr. Kenneth was suggesting. And usually there is a two-step testing process for the Lyme. That's what the CDC recommends. But the antibiotic treatment, which is the to-go, is the oral doxy, which is mostly effective for the mostly patients. But there were several studies which shows that the disseminated infection is not eradicated by the conventional antibiotics, like the tetracyclines or the doxys or the amoxicillins. So as a result, there is a macrolides, including the azithromycin, clarithromycin, erythromycin. They were recommended for a couple of patients who were intolerant to the first line therapy. So these are the basic antibiotics were available. But new mechanistic classes of antibiotics need to be developed to treat the infections, because there was very few are not able to tolerate to some of the patients. So we actually, with the help of our core facility, so we found that there is a almost we did almost 5,000 to 10,000 small drug, which we have a huge database. From there we found at least 15 to 20 molecules had a very strong potent. And among them, we found the disulfiram is in the top 5. And as Dr. Kenneth Lander already given a little bit background about the disulfiram, I don't want to dig in details about that. I just quickly go forward, as this is the drug which mostly used for the alcohol abuse, as it is an anti-abuse. As Kenneth Leonard had showed that this has beneficial effects in the treatment of the patients, both for the Lyme and the babesiosis. There was also there was an NIH study which is recently they're going to support to present the data on that, which we haven't seen yet. But the data is, but the clinical trials have been concluded recently. And if you see the most of the mechanism of action, how it acts, it is through basically it is an electrophile which loves the electrons. So it basically forms a disulfates with the thiol-bearing substances. And that's how it and Borrelia has a lot many different kinds of intracellular cofactors. That's how it goes and binds to them and basically prevents the cell wall synthesis. So that's how the mechanism we thought about the disulfiram to start with. And in addition to that, there is also a couple of thiophilic residues, which also disulfiram is known that it can be modified through forming the thiol disulfide exchanges, which evokes the antimicrobial effects. So if you see the disulfiram, it undergoes an extensive metabolism, which like initially it forms the DTDC, which can form the complexes with any of the endogenous metals like the copper or the zinc, and which can further metabolize it into the body. So basically, the physical discomfort which generally the patients experience, they consume the alcohol while receiving the disulfiram therapy, they are believed to be due to the collective inhibition of the aldol dehydrogenases by the metabolites 1 and 2. These are the metabolites 1 and 2, and which they further culminates in the toxic accumulation of the acetaldehyde. So that's how the discomfort has been experienced by the patients. So these are the couple of the clinical trials on the use of the disulfiram, not only on this was the Borrelia which was just recently concluded. There was also a couple of other use of disulfiram in HIV and the COVID-19. There are a couple of studies which have completed, and there is a very significant outpatient measures in all of them. Like in HIV, there is a fold change in the cell-associated HIV RNA in the total CD4 T cells. So they did it's not a monotherapy, they combined with an ART, and there is in a 3-dose course of, they used 500, 1000 and 2000 mix of disulfiram. So similarly, there is another study where they determine the effects of disulfiram on the pharmacokinetic of the other drugs, which is ritonavir and azatinib. Okay. Also, there was a WHO which has done a lot of clinical improvement with the COVID-19 patients. There are almost 140 patients. They also showed a significant impact of the disulfiram on those. So based on that, so as I said, we had done a lot of high throughput screening of the drugs. As I just told you before, like these are the low pack and the NIH, and then also Microsoft Spectrum and the Biomol. They're almost like 20,000 drugs. And then we used the bacteriotype flow assay, and we found like 150 inhibitory compounds, which were very much active for the stationary form of the Borrelia. And we determined the MICs and the MBCs, and we found the top 20 approved compounds, and which we published with both in 2016, 2017. So when I started my study, the disulfiram, as you know, it is not soluble in water. It's not readily soluble in water. So the only alternative we have was DMSO, but we don't want to do it in DMSO because DMSO at certain concentration is toxic to the mouse studies.
Pretty.
You gotta dial it down for the non-medical.
I don't. This is the medical. I'm not. I don't think this. I'm not sure who the audience was intended to be.
I was just gonna.
Okay.
Oh, the other crock pot, was that from the basement?
Matthew, your microphone is on. Can you put it off, please?
Oh, sorry.
Okay, thank you. So the cyclodextrin is the another solvent we had used, and this was recommended by one of our colleague in the lab, Dr. Mohammed, and he was like a chemist, and he was suggesting me because it's like a molecule with a cyclin structure where, because your drug is a small, so that it can go and hide inside, and it can slowly release by itself. It's like an organically, naturally, it can be possible. So I initially tried with different concentrations, then I end up like 10 to 20% of the cyclodextrin. That's how it completely soluble. The disulfiram was completely soluble. Then that's how I started using. And parallelly, I also dissolved the disulfiram in the DMSO, and both of them with different concentrations we tried in vitro and in vivo. So the in vitro data efficacy studies, we use the gold standard microdilution with both the log and the stationary phases of the Borrelia. And morphologically also be evaluated by the dark field direct cell counting, followed by the fluorescent microscopy using the cybergreen, which is for the green, which is for the live cells, and the propydium iodide, which is for the dead cells. So we had used both for the direct counting again. And finally, the best method is to use those colonies and then put it into the solid agar place. And after 15 days, you count them. That's like the solid plate agar method, which is where you can see the visible colonies. That's like these are all the 4 different methods we had used, and we validated our results. by using at least 5 to 6 different repetitions by the 2 different people. And then for all the experiment, what I'm going to show the data is doxycycline is our positive control. And we had used DMS1 cyclodextrin, as these are the solvents we had used to dissolve the disulfram. We use this as a negative controls. And this is the first, the microdilution method, where at the DSF, the doxy is in the red color and the blue and the green are the disulfram. In DMS1, the cyclodextrin, respectively. The green is the cyclodextrin DMS disulfram, and the blue is the DMS1 disulfram. You can see both of them eliminated almost 80 to 90% of the log phase spirocate, and it is around at 25 micromolar to the 0.65 micromolar. That's the MIC we had first found out. And you can see the doxy is the best significantly reduced to the viability is at around 90 to 97%. And these are the imaging of those pictures, where you can see these are the controls on the left side and the cyclodextrin DMSO. These are the negative controls. On the bottom of the doxy is the positive, and the disulfram with cyclodextrin at 5 micromolar. You can see very few were visible. And then the disulfram and DMS also very few visible at 5 micromolar concentration. So this is the log phase, and similar results we got at the stationary phase, which is like you culture them for 7 to 10 days, that's the log phase, and culture them for more than 3 weeks, that's how the stationary phase, and where you can see those round shaped, and then you can see the surrounding shape, like which is like the very like these are they doesn't look like like wavy shape. They look like more like a surrounding, like the round and the wiggling, very few wiggling sphere like this shape you can see with these. So again, almost 80 to 92% of the stationary phase persisters we compared with the controls, and we attribute this loss to the inadvertent effects arising from the colloidal films, because if you see the high concentration did not show anything, and at certain concentration which starts from the 25 almost 12 to the 6, where the low concentration showed a very strong inhibition. And we, as I said, like the reason what we attributed is because it could be the colloidal forms of these drugs, because as I said, when we are putting this drug, it forms into the small colloidal forms. And actually, we validated these results by using the DSF. There is a dynamic light scattering and electron force microscope imaging, where we found out those complex formations. And coming back to the pictures, like on the top are the negative controls. As you see, these are the congregate together, and there's like a little bit the shape also, the motility also will be reduced at this stationary phase. And you can see huge on the top, which are all the controls and the negative controls, and the bottom are with the doxy and the disulfram with 2 different solvents at 5 micromolar. And then you can see very few borrelia. And then finally, we did the solid agar method, and exactly the same results we could able to repeat in both the log phase and as well as in the stationary phase with the disulfram. As I said, the high concentrations, as you see, still have, we can see little bit more visible colonies, and with the dynamic light scattering, which is by the DLS analysis we did, and you can see there is a critical aggregate concentration, which is at this point, and we found that is around at 10 micromolar. And at above that, you can see a huge particle formation in both the DMSO and also production at this formation. And around this, the particle size was a little higher. So that could be the reason we didn't see very strong at the high concentration, but we do see a very good inhibition from the 25 micromolar until 0.62 micromolar concentration. And this was also followed by our AFM imaging of the DSF formulations. Again, at 100 micromolar, you can see these particle formations are pretty clear actually. So, and with DMS also, you can see those kind of particle formations. And then after the in vitro, we switched to the Lyme carditis model, mouse model, and we found there were a couple of mouse models are there. We compared with both the C3H and the like, which is a susceptible model, and there is a resistant mice. And you can see in the resistant mice, when we culture those Borrelia infected heart, we could able to see the presence of spirochetes in the B6 mice. So we end up doing all our experiments in the immunosusceptible mice, which is the C3H mice. And this is how the in vivo efficacy study was planned. We used the mouse model was C3H mice, and we used the 2 time points, which is day 14 and day 21. And after that, we given the antibiotics for 5 days consistently to develop the persistent infection, which is the Lyme carditis. And each group has a control which has no infection, and there is an infection group which has no antibiotics. There is an infection group plus antibiotics, which with the doxy and with the disulfiram. And we had tried with different concentrations of disulfiram, but we end up using at 75 mg, which is a little bit higher. And but this is equivalent to the human weight, which is 400 to the 500 mg per kg. So that's how the we end up using the 75 mg per kg for the mouse, and we use this through the IP route, which is intraperitoneal route. And then doxy was as a positive control. And this is the the tagline, the infection. As we do that day 0, we do the Borrelia infection. I do just behind the. This is the needle based doing the Borrelia around 10 to the power of like 10,000. The Borrelia will be introduced here, and we wait for day 14. And after day 14, we do the antibiotics for 5 days. And then after like 2 days, like 48 hours after the last dose, we terminate the mice, we collect the blood, heart, ear, and the bladder. Ear and the bladder, we culture them into the BSK medium, and we wait for 2 weeks and then we do the PCR to qPCR to check the levels of the Borrelia. And also for the blood, we check the total IgM and IgG levels. And for the heart also, we do the immunohistochemistry, and also we culture the like some or like half of the heart into the BSK medium and check their levels of the Borrelia after 2 weeks. Similarly, this is a day 21 group, and there is also day 28 group. The only difference is we extend this for day 21, where you can see the carditis from day 14 to the day 21. And again, we did the antibiotics after day 21 for 5 days and collected the same organs. And you can see with the disulfiram, these are all the studies done in the disulfiram not in DMS4. This is the cyclodextrin study we had done. The arrows, you can see this is the control and this is the infection group. The red group is the infection group in the ear and the heart base and the bladder. There is a large amount of Borrelia, almost you have 20,000 Borrelia in each. And with the dox and the disulfiram, it got reduced. They're not completely incomplete, but they're reduced at day 21. But in day 28, you can see very few Borrelia we could able to see by the day 28. So these are the total number of this is the qPCR data where you can see the number of the flag copies, which are the copies on the DNA. Almost like 70 to 20 Borrelia we found. Like these are the 5 mice I was showing. Like we did a couple of we repeated this with at least 10 mice. And then also this is at the day 28, where you can see very zero actually in couple of the cases, like very few in some mice there is no Borrelia too. And then we looked into the heart, and then we found inflammation, and which is like the carditis vasculitis. And you can see there is the percentage of the inflammation at the heart base. This is the control, and this is the PBS, where you can see the lot of the polymorphonucleosides, which are typically found at the lesion sites. And the disulfiram and the dox group has a very few the polymorphonucleosides. Which clearly signifies there is an inactive carditis than the infected mice. In most of the infectious diseases, the hallmark of any inflammatory tissue is basically the recruitment of these leukocytes, and the chemokines and cytokines also plays an important role. So from these, I isolated the RNA, and then I looked for the couple of the chemokines like the MYC2, which is an important chemokine for the macrophage attraction, and RANS, which is an important chemokine for the neutrophils. And you can see there is an upregulation of in PBS and the DOX group, and it is completely downregulated in the disulfiram group in day 21 and the day 28, which clearly signifies that there is a less amount of the macrophages and the neutrophils from our previous immunohisto data. This is kind of aligned with our previous data. Also, and we looked into the cytokines data, which is like the inflammatory cytokines, the MYC2, the TNFs, and the interferon gamma, and the IL-1 beta. And also we looked for the other protective macrophage, like the NOS2 and the INOS. The TNFs and the interferons, as you can see, was completely downregulated in the disulfiram group compared with the infection group. On the other hand, the IL-2 levels are also, the IL-1 beta levels are also very much controlled in all of them. So then we looked into the serum from the blood, and then we found out the IgM levels were a little bit low, but IgG levels were, there is no difference at the day 21. But we do see the subtypes of the IgG1, and where we find like the IgG1 and the especially the IgG1 levels were pretty low in the disulfiram group, but there is not significance in other subtypes of the IgGs like G2A and G2B and G3. There is no difference in among those, but very clear differences we found in the day 28 group, which is the little bit later group, where you can see the chronic or the persistent borrelia, where you can clearly see the disulfiram as very low amounts of IgMs and the IgGs. And also further, we looked into the subtypes like IgG1, G2A, and G2B. Also, they were critically low. The IgG2B levels were pretty much reduced. And we don't see much difference with the G1 and G2 because what we figured out that when we went on the literature, that the G2s and G2Bs, they mostly binds to the FCA receptors. So that could be one of the reasons where we're able to clearly figure out the differences. On the other hand, the G1s, they mostly they binds to the immunocomplexes. They found to the C1q, which is a complementary based mechanism. So that could be one of the reasons we don't see much differences among them. But the other approaches we looked into was there is a lymphadenopathy, which is in huge limbs, which you found at the day 10. This is a sham, which is a no infected and infected group. We find a huge difference. And in our groups also, when I checked the lymphadenopathy, the number of total lymphocytes were like almost 8 million. And then with the disulfiram group, they come back to the normal, almost 4 million, they come back to the normal. And this is like mostly characterized by there is an increased cellularity in this group, and there is a large IgM and IgG positive in these, like the plasma cells, and like the peripheral. Like I collected mostly the peripheral lymph nodes, the axillary, the branchial, and the cervical, the inguinal, all those 4 lymph nodes, and those are all very huge. And in the disulfiram group, you don't see any of them, like not normal basically. And that made me to got those lymph nodes, and I did the flow and then just did the immunophenotyping. And this was the gating strategy I had used. And then you can see these are the CD3 and CD19 and CD4 and CD8 cells. And from here, I found out the CD62L, which are the naive T cells, and CD44s, these are the memory T cells. And if you have only CD44, those are called the effector T cells. And if you express both of them, those are the memory T cells. And I'm just having the plots where you can see in the disulfiram group, the CD19, which is the B cells, were reduced, and also the CD3 cells, which is also reduced. On the other hand, the CD4s and CD8s were also normalized in the disulfiram group. Whether the CD4 cells has any ability, and these are the questions, as no one has done any detailed research about that, but whether they have any impact or not, because we looked into these are the naive T cells also decreased on the effector T cells are also decreased when compared with the infection group. So that clearly indicates there is an ability to enhance the antibody mediated immunity, which can cause the affinity maturation, and further they develop the long-lived memory T cells. So finally, I just want to quickly give the summary of the paper which we published, where we found that the disulfiram has an excellent Burrill cell activity against both the log and the stationary phase, that's in vitro, and in vivo, when we treat with the mice, it completely eliminated the Borrelia from the hearts and from the bladders by the day 28 post-infection. And it also showed a reduced expression of the inflammatory markers, and they were protected from the histopathology and the cardiac organ damage. And finally, the facts analysis where from the lymph nodes, they revealed and decreased in the percentage of the CD19 B cells, and there is an increase in the total percentage of the CD3 T cells and helper T cells, and the naive and effector memory T cells, and the disulfiram treated mice. So results from this in vivo study cannot be extrapolated to clinical practice at this point. We strongly believe that they form a strong basis basically for the future follow-up studies. and they promote the development of an effective formulations of the disulfiram for the clinical management of the either Lyme arthritis or the Lyme carditis or the neuroborreliasis. So these are the couple of publications which came out of from this. But I just want to give this last 10 slides where this is the new data which we haven't published. And this is the kind of some of the interesting data which we would like to move forward. As we saw with our in the last couple of years, we found there were a couple of limitations with the disulfiram. As we know, it's highly unstable because in the acidic environment and in the blood circulation, the half-life of disulfiram is just only 4 minutes. Okay, and it also belongs to the less water-soluble class drug with a probability of just only 0.5 mg per liter, or 0.2 mg per liter. So we found that the encapsulation of DSF into the nanoparticles, it can prolong its half-life and stability from the rapid deprivation in the bloodstream. So hence, when disulfiram is uncovered to the blood circulation, if you see the anti-alcohol potential, but their specific, like suppose the anti-borrelia or anti-cancer, that property is lost. It only stays for some time, but it's going to be lost because as the half-life is less and it can deplete right away. So we found a couple of articles where, like specifically the cancer related articles, and we found like they had used the nano-based like disulfiram delivery, and we thought that approach we could use it into the for treating the borrelia. So that's how I'm going to show a couple of the anti-cancer data, and then I'm going to show like how we have implied that into our borrelia research. So this was one of the study published recently where the disulfiram can reverse the resistance of some of the patients who are resistant to the cisplatin, like platinum-based drugs, because it basically blocks this multi-drug. There is a glycoprotein, it goes and blocks there. So in that way, those resistant patients, it can reverse the resistance to the anti-cancer drugs by basically inhibiting this P glycoprotein. This is called a P glycoprotein, which is in a multi-drug efflux pump actually. And it basically suppresses the activation of the further downstream NF-kappa B, which mostly causes the inflammation and which plays an important role in the drug resistance. And also, while inside, also it disrupts the DNA binding, and thus it prevents the further cell growth and the malignancy. So that's how the mechanism of the DSF occurs. But the approach was used the nanoparticles-based approach. These are the 5 different approaches people had been using, either using the lipid nanoparticles or the polymeric nanoparticles, or using the micellar, or the nanocrystals, or the microparticles. The advantage of these nanoparticles was their biocompatibility and their efficiency, and most importantly, they have the nano size, which basically makes them to enter and infuse into the cell membrane and to the vessels and to the different cell walls, and which takes them the passes into the different portions of the body until they reach to their particular target cells. So one of the very important recently there was one data which I thought I would share here is showing a very strong anti-tumor efficacy of disulfiram nanoparticles. Here they compared with the disulfiram which is in DMSO and disulfiram with nanoparticles in a human medulloblastoma intracranial xenograft mouse model. And in this mouse model, they had introduced the disulfiram and the disulfiram nanoparticles at 30 mg per kg. So with the nanoparticles loading, what they were given to the mice like in an IP route for 3 times a week. And they did the IVIS imaging. You can see the bioluminescence imaging. And you can see the results here. There is a significant decrease in this specific group in the tumor burden in the mice, but not with the disulfiram group. And also those were also, we can see here, by the day 35, the tumor size was very much reduced when you compare with the red group, which is the disulfiram group, compared to the purple one, which is nanoparticles. This made us and in addition to that, there is also these are all different kind of fabrication techniques, the compounds they had, lipid particles they had used, and the outcome they had got, like there is an improved in the tumor inhibition growth, or there is an specifically increased in the half-life of the disulfiram. Those are they really increased the betterment of the disulfiram. So that made us to do an very quick pharmacokinetic study, because we had made a couple of disulfiram coated. We used the oral based method. We used both the granulated formulation and also oral formulation, and the regular cyclodextrin which I had used for the. And you can see the cyclodextrin in 20 minutes in mice, it goes to the peak, and by 90 minutes, it goes back actually. But with the granulated oral one, when you can still sustains in by the 90 minutes, it is there. So this made us to we couple of optimize our nanoparticles, and these are the range of formulation parameters we had to find out. These are the couple of the drug polymer different approaches we do, the solvent evaporation and nanoprecipitation method, and these are the different which we had made. And we end up making 5 different formulations, six different formulations, and this is just the preliminary data. And I don't want to talk about those lipids what we had used. This is like Jay has all the different formulations. He has the one gave us the different idea behind the different formulations. And you can see with the formulations, when I tried onto the log phase of the Borrelia, you definitely has a very good impact. There is a strong inhibition starting at 1.25. It started at 10 micromolar and it kind of reached until the 1.25. It sustains for the long period of time. And also we got a good results in the stationary phase as well. But I'm not showing that data. But now we are in due course of doing in the in vivo study with the mouse model, like how we have repeated the cyclodextrin disulfiram study. So we are going to repeat the same study with these 4 different formulations. Looks like the combination of these formulations we are going to further enhance by adding some other 2 extra formulations. That's what we're still working on that. On that note, we looked into another approach, like the macrophages of the Borrelia. These are the bone marrow derived macrophages where we had given this is the control, and we had given the Borrelia here. And as I said, the NF-kappa B causes the inflammation. And from these bone marrow derived macrophages, if you treat with the disulfiram group, this is the last group, and you can see the disulfiram completely abolishes the NF-kappa B inflammation, where you have seen before also there is another cancer group which had showed. So this is also our one of the preliminary data, and also subsequent, excuse me, and also the subsequent downstream mechanisms are also inhibited, like the FAP4 is also inhibited, and we normalize the same for the actin. You can see the equal loading for all of them. And also there is an arginine which should be expressed in the disulfiram treated with Borrelia. So that's also is there in all the Borrelia treated samples. And that clearly shows that the disulfiram inhibits the NF-kappa B. This is one of the mechanism we found out. And also the GSK3, which is also plays an important role in the regulation of NF-kappa B. Those levels were also reduced. So those are the couple of other data which we would like to move forward. And also one more is about the RAF1 kinase, which is also plays an important role in antagonizing the caspase-1 during the pathogen-induced apoptosis, and that levels are also pretty much reduced in the disulfiram treated group. So this makes us have an important formulations in hand and also important mechanism. And further, only thing is we need to move forward with our in vivo data, and then we could able to again show with the formulations where we can able to increase the half-life, and further we can make a more efficient disulfiram molecule. So that's all I have for today. Thanks for your attention. Please let me know.
Hari, thank you so much. So I just wanted to brief for the benefit of many people here. So the issue with some of the problems researchers are facing with the disulfiram is they think, disulfiram, number one, is not a stable compound, number one. The number two is, in the area of drug development and pharmacology people, formulation people, they are well aware of this problem. Disulfiram is a notoriously polymorphic drug. That means, in the way we formulate, the efficacy will change. And also by aging, also the efficacy change. It's very, very tricky to formulate and to get a good data. That's the problem. So we wanted to address this problem. We wanted to find out why disulfiram is having a different behavior. And Hari Potala, some of my other group members, they spend a lot of time to understand at various concentration, both in vitro and also in vivo, whether any changes is happening. And there is any way to lock an effective conformation or effective polymorphic behavior, whether we can control them. That is our question. We have been working on that for the past 4 or 5 years. And, we are yet to publish the data. We are continuously repeating it. It appears by appropriately complexing, formulating with the lipids, lipid nanosomes. And we used all the FDA approved lipids for the formulation. It appears we can control the polymorphic behavior. That's what Dr. Hari Potala showed to all of you. And most of the data we presented, we are not clinician, we are not proposing anything as a clinical advice, but we just wanted to let you know that we put a lot of our efforts and then try to study this interesting molecule and also try to develop the best formulation possible. And then we'll be publishing it at one point, and then for the benefit of the audience. Thank you very much. If you have any questions, we have about 5 to 6 minutes. Please consider asking questions to both Dr. Kenneth Legner and also Dr. Hari Potala. You can answer, and you can ask the question, and I can definitely intervene in case if I need to be, answer the questions. Thank you all.
Yeah, it's me. Hi, can you hear me?
Yes.
Perfect. Hello. So thank you for the nice talk. My question is about the pulse dosing with higher doses of disulfiram, because I've seen you mentioned the study in HIV, and in this study they tried to measure the blood concentration of disulfiram, and only in this very high dose group, like in the 1,000 up to 2,000 milligram per day, they were able to come over the MIC, what you have done in your vitro studies. And because I've seen a lot when patients, they stay very long time on this drug, and I think this can be very problematic. So my question is one to Kenneth Legner, if you have seen when he pulse dosed this drug, if you ever seen any problems with the dopamine, because some people have, including myself, had problems when I came off the drug, that maybe when you only take it for a few days, there's not that problem to stop it cold turkey, or that's one question. The other was with the high dose of disulfiram, is can it be that the drug is more in the tissues and it's not able to measure it in the blood, or it's because it's weird sometimes when I read the studies, I was thinking, well, how this can be working when they're not able to measure any concentrations, and
I have to say that in terms of pulse dosing, that's restricted to an N of 1. This one gentleman who was in the first 3 patients, and also he did have problems when we first used it on him, and he had a syncopal episode. Luckily, he wasn't seriously injured, and that was probably related to some of the other pharmacotherapy that he was on for his depression. And so we made sure to take great precautions and modify his medications. But again, this is one case, and this is a gentleman presently in his mid-80s but he's tolerated it very well. And I think that trying to measure disulfiram in patients after doses, the question is, what are you measuring and how are you measuring it? Well, the only thing I can say is this approach in an N of one patient, and before we applied this method, he was having lots of problems, and he's been very, very stable for, I guess it's more than a year, I mean, probably more close to 2 years. Good quality of life, and able to do most of the things that he wants to do, and he still is under concurrent psychiatric care for those issues. But in terms of his general status and his functioning, he's quite satisfied. And I don't know. Can I explain it? All I know is that it seems to work clinically.
Yeah, I mean, I've seen this in my case too. This was the first time where my IgGs went down and everything. But no, I was thinking about the study because in this HIV study they used only for 3 days very high doses, and there were no serious side effects, and they had a really high concentration then of disulfiram in the blood for a few days. So that I was thinking maybe that's something not too bad that you only take it for a few days, for 4 days, and wait. Who knows?
Well, you want to try to maximize the effectiveness and minimize or avoid, if possible, toxicity.
Exactly. Yeah.
I'd just like to comment to Dr. Patula. I'm so impressed by the work that you've done. It's like incredible. So kudos.
Yeah.
Thank you, Dr. I appreciate it.
Can I only ask one question about the nano particles of the disulfiram? When you made this new formulation or are trying to stabilize it more, when you reformulate these drugs, do you need then clinical trials for that to get this approved, or is this something like you change a little bit of the drug and you still can use it, or you need a completely new FDA approval for that?
Yeah, let me answer this question. So it's for the approval authorities to decide whether they need, anybody wanted to take this license, this formulation from, the university and then try to develop it. It is for the authorities to decide whether they want an additional data or additional clinical trial. We are not having any power to decide on that.
Okay.
And also
Thank
to
you.
add to that, the components which we are using, like PLG or whatever those are all FDA approved. So there won't be only the combinations, what Jay said, that could be one of the things we need to consider with the scrutinizing committee. But the materials what we are using, those are good to go. Those are all FDA approved materials.
Oh, okay. So maybe you don't need directly something.
No,
Because I've seen.
I'm sorry, So any formulation or anything in the route of delivery or a formulation, it needs to be approved. It is not like, we used all the FDA approved, entities and we mix the drug and then we compound it. We can't take it for granted. It needs to go for an approval, yes. And most of the discussion that is happening, except, Dr. Kenneth Legner is a clinician, he's a licensed medical practitioner. We are not. And whatever, we say, it is just whatever we observed in our animal work and also in vitro work. And it needs to be validated by clinician and, appropriate authorities. It should not be taken as a medical advice.
Okay.
Thank you so much for all of your participation. We are planning to have this program every month. We call it as a BioNeomed Tech Talks. And Chris, you can conclude it. Thank you so much for all of your thing. And especially I take this opportunity to thank the speakers, Dr. Kenneth Legner and also Dr. Harihara Potula. Thank you. And all the group people who are involved in the work for several years, and also the funding that we received from specifically from Bay Area Lyme Foundation and also BioNeomed and other funding agencies, and also donors, they provided money for this research. Thank you all.